Hi, I'm Stuart Worboys, a botanist with the Australian Tropical Herbarium based at James Cook University in Cairns. This presentation has been put together in collaboration with Barbara Waterhouse of the Northern Australian Quarantine Strategy with a goal of introducing vets to techniques for plant sample preservation and identification. So as botanists, we're really keen to ensure the specimens sent to us for identification are of the highest quality. So if you follow this guide, you'll make us happy when you send stuff in. So to set the scene, I have a small story from the intersection between botany and vet science. It starts in 1970 in the Daintree area north of the Daintree River. Grazier John Nicholas suffered the loss of several cattle over a period of days. The symptoms displayed by the cattle were consistent: powerful muscle contractions and excitement, followed by collapse and death. Symptoms similar to those caused by strychnine poisoning. So the grazier was convinced that they were being poisoned, so he called in the police. The police took one look and then called in the government vet at the time, Doug Clegg, who conducted an autopsy. In the stomachs of the cattle, the government vet found partly masticated remains of large seeds, which he traced back to their origin tree. He collected specimens from the tree and sent them off to Stan Blake, the head of the Queensland Herbarium, who was fortunately in Townsville at the time. So using the specimens sent, Stan Blake recognised the plant as a species not seen for 70 years and actually long thought extinct. He went as far as declaring the botanical find of the century, but later he was able to collect more specimens, including flowers, and using those he described the plant as a new genus, Idiospermum australiense, in recognition of its idiosyncratic enormous seeds. It's the only member of its family in the southern hemisphere, and it probably has the largest seed of any plant in Australia apart from the coconut. This whole story of botanical discovery only began because quality botanical specimens were collected, and that's what I want to discuss today. The physical process of collecting and preserving plant specimens has changed little in centuries. It's very low tech. At the Australian Tropical Herbarium, we hold collections made by Joseph Banks and Daniel Solander in 1770. And because they were well collected, well preserved at the time of collection, and well maintained for over 250 years, they are still in excellent condition. These specimens represent both important historical documents and valuable scientific records of the existence of a particular species in a defined place at a defined time. So let's run through what needs to be done to collect botanical specimens that will last, are useful for both for identification and as species records. To confidently identify a specimen, the botanist needs leaves, flowers, and fruits or seed heads, and also an idea of what the plant looks like in its habitat. So before you go collecting, take a photograph of the specimen in its habitat, and also get some close-ups of flowers, leafy branches. If there's fruits, take photos of those, and take a number of photos from different angles. An example of why this is important is flowers. Beneath a flower, you have a structure called the calyx, and if you just have a photo of the top of the flower showing its petals, we won't be able to see the calyx, and it's an important feature for identification. Collect a leafy specimen around about 50 centimeters long minimum, and tag it. On that tag, write a unique reference number, and that reference number will also be given in your field notes, and it'll also refer to the photographs that you've taken. Press the specimen immediately, or if you can't press it, keep it cool, not frozen, and damp until you can. So just treat it like a lettuce that you've just bought from the supermarket. Wrap it in wet newspaper, chuck it in the esky or a cool room or the fridge, and keep it in good condition until you can press it. One additional thing you can do just before you press it is to scan it on the photocopier, and the scan looks something like this.
The specimen data is critical. To start off with, give the specimen a unique code that you can reference in your notes, and all parts of the plants that you collect should be given the same code. So if you're picking fruits up off the ground, give them the same code as well. That code is up to you. It could be your initials combined with a date, or your initials plus a number in a sequence, or just Jerry's farm plant number 3. All the notes and photos that you take subsequently must reference that code. So, in your notes, you should be recording the following information. Firstly, your name and the unique collection code, the date of the collection, a field name. So a field name might be the common name that provided by the landowner, or just use your own name that you make up on the spot. For example, yellow flowered pea or sticky leaf herb. This provides a useful informal reference for later discussion. The location, give a good plain language description, and latitude and longitude. Latitude and longitude can be obtained from most smartphones. A site description, for example, was the plant collected in open pasture? Was it on the roadside, in natural bush, growing in the creek, or a weed growing next to the cattle yards? What's the soil type? What's the slope? What's the associated vegetation look like? Put down the reason for collecting. For example, it was a plant suspected as being responsible for stock poisoning. And finally, a description of the plant itself. Record features which will not be evident in the pressed specimen. For example, is the specimen from a tree, a shrub or a vine? What's the flower colour? What's the texture of the bark? What's the plant's height? Do the flowers or the crushed leaves have a distinctive odour? You don't need to record the obvious stuff. The botanist will know that the leaves are green.
Here's an example of a botanist's field book. These days our locality information is a little bit more precise with the aid of GPS, but the information collected is largely relevant to this discussion. So, pressing the specimen is pretty straightforward, but a well-prepared specimen will last for centuries. Firstly, trim your specimen to fit inside a tabloid newspaper, just 2 or 3 sheets of newspaper enough. Sandwich it between sheets of corrugated cardboard, and squash the plants with bricks or books or a plant press. Keep under pressure in a warm, airy place, and change the newspapers every 1 to 3 days until the specimen is dry and crispy. If your specimen has large fleshy fruits or lots of flowers, you can pickle them in alcohol. Ideally, we'd want 70% ethanol, but a mixture of 70% metho and 30% tap water will be enough. Or if you've got some overproof rum, chuck it in overproof rum. That will preserve the specimens. You need high alcohol content. Ensure a copy of that unique collection code goes into the jar so you don't lose track. When you're sending this specimen, remember that you can't post flammable liquids. So you just have to decant the liquid and send the wet specimen in the post. So here's a photo of a botanist's press. You can see all the corrugated cardboards, and you can see some little plants sticking out. Now, you don't have to have this level of technology. You can use fridge shelves, you can use planks, anything large and heavy and rigid that will press the plant and allow it to dry. And this is what the setup will look like. You can see here there's a specimen which is large enough to fit inside a regular newspaper. Here's another specimen here. Here's a pickled specimen. Here's the botanist's notebook. So the next step in the process is to get your specimen identified. Any information that you provide with regard to common names is always helpful to us. So a field ID is helpful. You can, if you recognise the plant as a yellow flowered pea, then please put that in. That information is really helpful. A lot of people ask questions about DNA analysis of stomach contents. It is possible, but it's costly in both time and money, and may not provide the clear results desired. Expert ID of dried specimens is the preferred option. That expert ID can be provided by either state and regional herbaria, who offer identification services, usually for a charge. There's also a few people, including NABS botanists, Northern Australian Quarantine Strategy botanists, who are willing to help out with plant identification. So those resources are in the handouts accompanying this. This is the location of the various state and regional herbaria. There's one in Perth, Darwin, Alice Springs, Cairns and Brisbane. So these are the state and regional herbaria who offer plant identification services in Northern Australia. The Queensland Herbarium, the Australian Tropical Herbarium, Perth Herbarium, Northern Territory Herbarium based in both Alice Springs and Darwin, and NARC staff, the Northern Australian Quarantine Strategy staff, have also put their hands up to assist. More details about how to contact these institutions and their physical locations, postal addresses, are provided in the handouts. Thanks for listening.